Hi, I'm Spencer Krieger, and I'm excited to share work I did with my PhD advisor, John Kasesioglu, titled Computing Optimal Factories in Metabolic Networks with Negative Regulation. Factories are a representation of pathways, which are a collection of biochemical or physical reactions that culminate in a specific cellular response. A simple example is the insulin pathway, which results in the storage of blood glucose in the cell. First, insulin docks in the insulin receptor, which causes a cascade of internal reactions, opening the glucose transporter. This allows glucose to be taken from the bloodstream into the cell, where it can be stored as glycogen or converted into fatty acids. A metabolic network is a collection of reactions from many different annotated pathways, all concatenated together. Here I have a picture of a metabolic network that is a combination of gene regulation, cell proliferation, and apoptosis pathways, with receptors around the outside of the cell acting as sources. For many reactions in a metabolic network, the stoichiometries of each participant in the reaction is also recorded, and a collection of metabolic networks from many different organisms is called a network database. In this presentation, we present solutions to the pathway inference task, which is, given a set of source compounds, think of things present around the cell, and a set of target molecules you would like to make, we want to find the series of reactions that produces the targets from only the sources. Why do we care about inferring pathways in metabolic networks? Well, pathway inference can identify crosstalk, which I explain in this picture. So pathway 1 is an annotated pathway from S to T, and pathway 2 is an annotated pathway from S prime to T prime. By inferring a pathway from S prime to T, we show that the activation of pathway 2 can cause partial activation of pathway 1. A second purpose of pathway inference is identifying therapeutic targets. Given a disease symptom T, we can infer a pathway from our sources, which identifies a protein represented by vertex V as a potential therapeutic target for treatment of the disease. These metabolic pathways are composed of reactions. The general form of a reaction I give here, with the reactants on the left, which I've colored in blue, the products on the right, which I've colored in pink, and these small letters represent the stoichiometry of the reaction, which are positive integers that tell you how much of each substance is used in the reaction. Reactions can also have regulators, where positive regulators promote a reaction or allow it to happen, and negative regulators inhibit a reaction or stop it from happening. A reaction will only proceed when all reactants are present, and it will produce all of the products. So to infer pathways, we would like to represent these reactions as edges in graphs. So given this reaction that I showed you on the previous slide, traditionally been represented in ordinary graphs, where each reactant or product is represented by a vertex in the graph, and each reaction is mapped to multiple edges, where you take one edge between each reactant and each product. Traverse the edge from A to C, there's no constraint on B being present already. We instead want to model these reactions in hypergraphs, where we can model the entire reaction as a single hyperedge, with the reactants in the tail of the hyperedge and the products in the head of the hyperedge. Models that all of the reactants must be present for this hyperedge to be traversed, and upon its traversal, all of the vertices in the head are considered reached, and those products are produced. I will now introduce the concept of metabolic factories, which are a hypergraph model for pathways and specify a flux for each reaction in the network. Flux is a relative usage of a reaction in a metabolic network and is represented by a non-negative real value for each reaction. A hyperedge active if it has a non-zero flux for its reaction. We store stoichiometries in the stoichiometry matrix M for each metabolite in each reaction. For all intermediate metabolites, we specify that the metabolic flux must satisfy either that the influx, which is the flux on the hyperedges into the vertex, is equal to the outflux, or the flux on the hyperedges or reactions leaving the vertex. And we constrain under conservation that these are equal by this equation. Or under accumulation, we constrain that the flux in is no less than the flux out, which is given by this inequality. Finally, a factory specifies a flux on each reaction in a network that produces all targets from the sources and satisfies either conservation or accumulation for all intermediate metabolites. To give some related work, prior pathway models have also used hypergraphs. In particular, minimum source factories have also found factories, but where they try to minimize the number of sources. Another hyperpath model are shortest acyclic hyperpaths, where hyperpaths don't capture stoichiometry and therefore don't accommodate accumulation or conservation. Our new pathway model are minimum hyperedge factories, where we seek to minimize the number of active edges in our factory following the principle of parsimony. Also incorporate negative regulation into our factories, where negative regulators inhibit reactions. cartoon shows a reaction proceeding normally, where the substrate binds to the enzyme in the active site, which causes the enzyme to then release the two products of the reaction. The bottom row shows inhibition, where the inhibitor instead blocks the active site, which causes competition with the substrate and inhibits the reaction from taking place. Prior pathway models do not consider negative regulation, which we consider for the first time. I will now preview our contributions. So we've introduced the new problem of finding a minimum hyperedge factory that uses the fewest possible reactions. We proved that minimum hyperedge factory is NP complete, and therefore there is likely no method that finds optimal factories that is efficient in the worst case. We also consider negative regulation for the first time, considering both first and second order effects. We performed the first comparative study of hypergraph pathway models, comparing minimum precursor or minimum source factories versus shortest hyperpaths versus minimum hyperedge factories, considering both first and second order negative regulation. As demonstrated through comprehensive experiments across all instances from standard reaction databases, our approach is remarkably fast in practice, with a median runtime of only a few seconds, while including both first and second order negative regulation. We have implemented our approach in the tool Odin, which I'll show at the end of the presentation where it's available. To move on to our methods, I will first formally define the minimum hyperedge factory problem. Its input is a hypergraph G, a stoichiometry matrix M, which records the stoichiometries for each reaction, the candidate sources S, and a set of target molecules T, and also a minimum flux constant epsilon, which is greater than 0, which we will use to enforce non-zero flux for all targets. The output is the flux such that for intermediate metabolites, either conservation or accumulation holds. Every target is produced with flux at least epsilon, and the number of active hyperedges is minimized. I will now introduce our two orders of negative regulation, which consider both direct and indirect effects. So first order negative regulation considers direct interference within reactions inside the factory. Given a factory, as I've drawn here, we call it first order negatively regulated if a vertex in the factory, here R, regulates another edge in the factory, which would invalidate the factory. For second order negative regulation, we consider indirect effects, so interference from outside of the factory. Here we consider the active sources S and S prime, and if a vertex R is reachable from the active sources that negatively regulates an edge inside the factory, we consider this factory invalid because we know that the sources are active, so R could be created and inhibit the factory. This leads to the minimum hyperedge factory under zeroth first and second order negative regulation, where zeroth order negative regulation simply ignores negative regulation effects. We have proven that all versions of minimum hyperedge factory, even including negative regulation, and under accumulation and conservation, are NP complete. I will now introduce our method to solve the minimum hyperedge factory problem. So we reduce minimum hyperedge factory to a mixed integer linear program. MILPs generally are composed of variables that can be real or integer valued, constraints on these variables that are linear inequalities, and finally, an objective function, which is linear in the variables, which is generally minimized or possibly maximized. Our specific MILP for min-edge factory has two variables for each hyperedge in the hypergraph. First is a real valued flux, f sub e, that is between 0 and 1. And the second is an integer valued active edge indicator, x sub e, also between 0 and 1, which will show whether the edge is active or not. For constraints, we have active edge constraint. Active edge indicator is equal to 1 if any flux is on a given hyperedge. Either conservation or accumulation constraints for each intermediate metabolite, and target constraints, which for each target ensure that it is created in a quantity at least epsilon. The objective function is to simply minimize the number of active hyperedges, which minimizes the sum of these binary active edge indicator variables. Now I want to describe how we incorporate negative regulation into our MILPs. For first order negative regulation, we add a constraint for each hyperedge E that is regulated by a product of hyperedge F that ensures that both E and F cannot be active. So here we take the binary indicator variable for both E and F and ensure that their sum is less than or equal to 1. Tackling second-order negative regulation is a little bit more complicated. Here we solve a series of MILPs that compute first-order factories satisfying second-order negative regulation using the fewest reactions. At a given iteration I, we solve the current MILP to obtain flux. Then, if an inhibitor of an active edge in Fi is reachable from its active sources, we add a constraint ruling out Fi and loop back to iteration I plus 1. We do this because this would mean that an inhibitor is reachable from the active sources, which would mean that our solution, Fi, does not satisfy second-order negative regulation. If this is not the case, then we return Fi as it is a valid factory that satisfies second-order negative regulation. Now move to experimental results. Hypergraphs from human-curated metabolic networks from standard databases, and I will now give a concrete example of how we would map the formation of this complex, A/B/L, to a hypergraph. We represent each protein and small molecule, like ligand L, as a vertex along with the ABL complex. We then map each reaction, like the one given here, to a hyperedge. So this reaction maps to this hyperedge. We built these hypergraphs over the standard metabolic networks of Reactome, which is a large network database of human pathways, and then metabolic networks of organisms from MetExplore, which include these 7 organisms. Larger hypergraphs are, they can have up to 20,000 vertices, 12,000 hyperedges, where 500 of these are annotated with negative regulation, and up to 8,000 sources, where sources are all vertices with no in-edges. With all of the hypergraphs combined, we end up with 6,000 targets, which are all vertices with no out-edges. Hyperedge factory is to find a factory from the 8,000 sources to a single one of the targets, so we have about 6,000 instances of the problem as well. Our results, where we compare different pathway models on the same instance. We found the minimum hyperedge factory, the minimum source factory, and the shortest hyperpath. We compare pairs of models over fractions of instances where both solutions meet some criterion, factory models, and over the organismal datasets, which is all 7 MetExplore datasets and Reactome. The fin determines whether min edge factories have a minimum number of sources. So here on the organismal datasets, we see that on over half of the instances, the min edge factory does have the minimum number of sources, and Reactome it's about a quarter. On the other hand, if we look at the number of instances where the min source factory has the fewest number of hyperedges, we see that on either dataset where the min source factory is edge optimal. Compare the number of instances with the minimum edge factory that also has a hyperpath. So here we see that there are many instances on both datasets for edge factory, but no hyperpath. If we look at this now the other direction, there are no instances that have a hyperpath, but no minimum hyperedge factory. So the big takeaway from this table is that the minimum hyperedge factories are often optimal for other models, but this is not the case in the reverse. I next want to show one biological example and give you the minimum hyperedge factory, the minimum source factory, and shortest hyperpath on that example, and then compare and contrast these different pathway models. So here is a minimum hyperedge factory from these sources that I show here at the top to this target, which I show in the bottom right. I'm going to first contrast this minimum hyperedge factory to a shortest hyperpath, where shortest hyperpaths are a ring of hyperedges, so that for each hyperedge, all of the things in its tail must be covered by the heads of previous hyperedges. So in this situation, this vertex, NADH, before it's in the head of another hyperpath. So the shortest hyperpath replaces this hyperedge with these 2 hyperedges, and therefore the shortest hyperpath is 1 hyperedge longer than the minimum hyperedge factory. For the same instance, here's the minimum source factory, that although this factory uses fewer sources, it uses many reactions that don't really contribute to creating the target. And then also on the left part of the figure, you can see this useless cycle that does not use any more sources and therefore has been included even though it has more hyperedges. So the big takeaway comparing minimum hyperedge factories to minimum source factories is that minimum hyperedge factories will have no useless cycles and will create better pathways because they minimize the number of reactions. Next, I'm going to compare zeroth first and second order negative regulation, where zeroth order does not consider negative regulation at all. Reactome is the only database where regulators are annotated. The first comparison compares how many instances where the first order optimal solution is not zeroth order optimal, meaning the first order factory that has no direct interference and that does not consider interference from negative regulation. This second row considers the same thing, but optimality is now with respect to the number of sources in the solution. This third row now considers optimality with respect to the number of hyperedges, but compares second order instances to instances under first order negative regulation. For all of these instances, ignoring negative regulation means you return a non-viable solution because all optimal zeroth order factories have interference from negative regulation. Here, I show the zeroth order factory on a concrete example, where ellipses have been drawn in place of hyperedges up to sources that are the same between the first order factory and the zeroth order factory, and the target is given in the bottom right corner. Here, GSH inhibits its own reaction, and therefore this factory is not viable under first order negative regulation. Instead, the first order factory replaces this hyperedge with a series of hyperedges shown here on the left. Here is an example that shows both of these factories. Takeaways from this example are that first, the first order factory has 4 more hyperedges than the zeroth order factory. And second, the first order factory avoids this inhibited reaction and therefore produces a viable factory. Next, I want to talk about the running time of our implementation of our methods called Odin. The median runtime of Odin is only 3 seconds, even including both orders of negative regulation. The maximum runtime under zeroth and first order negative regulation is just over a minute. For second order negative regulation, it can take much longer, up to 70 hours. However, this only happens on 11 of the instances that take more than 6 hours, and this only occurs when no second order factory exists, which means we end up enumerating all minimal first order factories before finally returning that there's no solution. In conclusion, we introduced the new minimum hyperedge factory problem and proved it was NP complete. We incorporated negative regulation for the first time into pathway inference, showed that we can compute optimal factories remarkably fast in practice. And an implementation of our method along with all data sets is available at odin.cs.arizona.edu. We have a couple of ideas for further research on this topic. We would like to extend our orders of negative regulation to consider third order negative regulation, which would model mutual interference between regulators. But this requires modeling reaction rates or order of reactions. We would also like to automatically determine a valid value for the minimum flux constant, epsilon, which is a parameter of the model that must be set and is currently set manually. However, the wrong value of epsilon may exclude valid factories or cause numerical issues for our solver. We would like to thank Tia Morali and Anna Ritz for introducing us to this problem and for discussing the data sets with us. And we would like to thank the National Science Foundation for funding. Thank you for listening to my presentation.